to introduce you to Dr. Mahendar Thudi. He is working as a visiting scientist in our green legumes research program at Center of Excellence in Genomics, and in collaboration with several colleagues from ICRISAT and from outside. For last 1.5 years or so, he has been working on this particular project. We call it association mapping for abiotic stress tolerance in chickpea. So I think now I would like to invite Mahendar. So Mahendar, floor is yours. So please welcome Mahendar, and then you can start.
Thank you, sir. Thank you for your kind introduction. Good afternoon, everybody. Today I will be talking on association mapping for abiotic stress tolerance in chickpea. This work has been carried out in collaboration with Drs. Upadhyaya, Abhishek Rathore, Puran Gaur, L. Krishnamurthy, Manish Rudkiwal, Spurti Nayak, and Sushil Chaturvedi from IAPR, NVPR Gangarao from ICRISAT, Kenya, and Asnake Fikre from EAR Ethiopia, Paul Kimurto from Egerton University, Kenya, and Prakash Sharma from Indraprastha University, Delhi. So I have divided my topic like I will be first discussing on the linkage analysis and association mapping. Why should we do association mapping? What are the kinds of association mapping? And then the results of my study. For long being, breeders and geneticists are interested in understanding the genetic basis of complex traits like drought tolerance, salt tolerance, flowering time, yield, etc. Basically, in linkage, they were employing the linkage analysis. In this linkage analysis, we were basically using biparental mapping populations. There is a disadvantage using biparental mapping populations because sometimes the number of polymorphic markers on a given population between a given parental combination may be very low, and the given parental combination may segregate for only a given trait of interest. And in linkage analysis specifically, we will be looking at two alleles, maximum of two alleles and few recombination events that are going on to compute the genetic distance between markers that are in linkage. Unlike linkage analysis, here in case of association analysis or association mapping, we can determine the polymorphism even in a single gene. This is possible because of historical recombination events that are occurring in the natural populations. In case of linkage analysis, we will be using the structured mapping populations, but in case of association mapping, we are using the natural populations like germplasm collections or reference set like that. So in order to understand the complex traits, we are going for QTL analysis, and here is the mapping resolution that will be achievable. The QTL resolution that is achievable using different mapping populations like double haploid mapping populations, wherein the gametes are turned into double haploids by treating with colchicine. Here we achieve only about 10 centimorgan of QTL resolution at large score 5. However, in case of RIL mapping populations wherein the recombination events are more, this is possible for achieving up to 5 centimorgan QTL resolution. But in case of association or diverse panel, where historical recombination events are taken into consideration and we can trace back the allelic history of each allele, and it is possible to achieve a resolution of 1 centimorgan.
Sorry, I'm on the talk right now.
Sorry. So why should we take up association mapping? Association mapping helps us in fine mapping of QTLs, validation of candidate genes, and we will be able to detect polymorphism within genes that are responsible for different phenotypes, and we can also determine the haplotype blocks that represent different alleles of a given gene, and it helps in marker assisted selection, and we will be able to understand the population genetics as well as the evolution of a particular population. Basically, there are two kinds of association mapping, that is genome-wide association mapping and candidate gene-based association mapping. In genome-wide association mapping, we will require large number of markers, and in candidate gene association mapping, we require only small number of markers very near to this candidate, related to the candidate gene. The success of either methods depend upon the population size as well as the degree of linkage disequilibrium in the population. So when we compare the genome-wide association and candidate gene-based association mapping, as I mentioned earlier, genome-wide association mapping requires large number of markers, and this involves cost. And candidate gene association mapping is basically like low cost and a hypothesis driven and trait specific approach. But here we will be missing the alleles which are unknown. And like in case of association mapping, we should have a germplasm, and this germplasm needs to be phenotyped, and the germplasm can be genotyped for genome with markers, large number of markers for genome-wide scan, and we can also genotype for a specific candidate gene, and we can determine the population structure as well as kinship relationships, and then go for association analysis. So basically, linkage disequilibrium determines the number of markers required and appropriate mapping strategy. So when we have when the linkage disequilibrium decays slowly, we require a small number of markers, and when the LD decays quickly, we require large number of markers to go to the target gene. Basically, there are 5 steps for association studies. First, we need to select a diverse panel for association mapping. It can be a group of individuals from a natural population or germplasm collection. We should go for precise phenotyping data generation and go for either candidate gene or high density marker genotyping on the panel, and then go for determining the structure of the population and the kinship relationships, and go for association studies. So in case of Chickpea, we have reference set as well as mini core collection. Mini core collection is a part of a reference set, and we would like to know whether the reference set is a good can form a good association panel or not. For that, what we have done, we have used SSR markers as well as SNP markers. We have used 36 SSR markers and 791 SNP loci. So we could not find any specific grouping either based on their geographic origin or based on the market class or any biological status of the species. In case of SSRs, though there is little grouping, so this panel is most suitable for association studies. So in order to determine the population structure, we have used structure software, wherein we have used calculated the number of subpopulations from 2 to 14 with 100,000 replications and 2 iterations and 200,000 MCMC replications. When we plotted the delta K against delta K, we have found that there are 3 subpopulations in this particular reference set, and this is the genome composition of the 3 groups. What we have, the 3 subpopulations are 3 groups that we have determined. Here we have used all 1157 dart loci what we have, and constructed the neighbor joining tree. The green ones represent the Desi type and red ones represent the Kabuli type, and the black and blue color genotypes represent the wild as well as pea-shaped chickpeas. So this dendrogram also indicates that there are three subgroups or subpopulations in this reference set. The phenotyping data was assembled from different collaborators like Drs. Hari Upadhyaya, Puran Gaur, Krishnamurthy, Chaturvedi, Gangarao, Asnake Fikre, Paul Kimurto. So we have phenotyping data for 3 years and 3 replications for root traits. For transformation efficiency related traits, we have 2 years data for 3 replications at Patancheru, and yield and harvest index related traits for 2 years from 3 locations in 2 replications, and heat tolerance data for 1 year in 2 replications from 2 different locations, Patancheru and Kanpur. And mini core collection, we have 7 season data for different morphological traits and yield component traits from Patancheru. So we have more than 35 different traits that we have phenotyping data, and these are the root traits that we have phenotyped, and this is for 3 seasons, and the morphological traits vary from 1 season to 14 season data, and yield and harvest index related traits around 10 season data we have, and for carbon isotope discrimination data we have 2 season data, and for SPAD chlorophyll meter readings we have 2 season data. So we have more than we have around 2000 or 957 marker data generated on this reference set, 36 SSR markers and 1157 dart loci, 651 SNP markers, and 113 SNPs based on the genes, 10 based on 10 genes, and we have used 1872 markers for our association studies. The marker attributes for all the markers were calculated like gene diversity, heterozygosity, and polymorphism information content values for all the markers systems that we have employed in for association mapping. In case of for establishing marker trait associations, we have used Hassel 4 version, and the genotyping data like for SNP data we have used the same like ATGC, whatever the nucleotide information that we have, and dart markers like 01 presence absence scoring data, and SSR's allelic data. We have employed optimum compression model with P3D and in MLM analysis. Generally, MLM analysis takes thrice time that is required, that is equivalent to the number of genotypes. But by employing the P3D and optimum compression model, we can reduce the time of computation heavily. To consider a marker trait association to be significant, we have used the p-value. We have considered only the marker trait associations above this threshold, like 10 power minus 4. And we have used haploview for determining the LD blocks. As a result, we have we could establish 583 marker trait associations, and these are the marker trait associations for root traits. Root surface area is having the marker trait. There are 4 markers that are associated with root surface area, and this is explaining. These markers are explaining around 4.7 to 17.3 phenotypic variation, and the p-value is also high. And in case of morphological traits, basal secondary branch as well as basal primary branch are explaining higher p-values, and the phenotypic variation was high in case of basal secondary branches. In case of phenological and yield related traits, we have the days to flowering and days to maturity are explaining almost similar phenotypic variation, and we have maximum number of marker trait associations for 100 seed weight. There are 30 markers associated with 100 seed weight, and marker trait association were 113, and these markers explained about 38.5% phenotypic variation, and the p-value is also significantly high compared to any other marker trait associations that we found. In addition, we have also had marker trait associations for transpiration efficiency related traits. In case of specific leaf area, this was explaining about 22.8 phenotypic, 4.6 to 22.8 phenotypic variation, and the p-value was 5.17 to 17 into 10 to the power of -5. So what is the use of this marker trait associations that we have established? Let us see, like for root length density, when we have when we select for this marker, which is on LG2, and this is at linkage, this is at 15 centimorgan position, and the major allele is T, and this contributes about 0.03 in terms of root length density. And let us say in case of yield, there is one gene-based marker, ASR, and this is not mapped onto any linkage group, but however, this is having the major allele as G, and this contributes about 764 kg per hectare higher yield if we select for this particular marker, for this particular trait. Similarly, in case of all the marker trait associations that we have identified, in case of delta carbon, all were showing only negative associations, and this is one of the marker that is showing high negative association. So these are different marker trait associations which can be used in breeding. These are Manhattan plots drawn. So this is the linkage. These are linkage groups, distance in centimorgan, and this is the negative logarithm of p-value. So this is this particular marker which is coming on linkage group 4 is having highly significant association with root length density of this particular observations of this particular year. Similarly, for yield under irrigated conditions, we have one marker on linkage group 4, which is having significant association. And in case of harvest index under rainfed conditions, we have four markers on linkage group 6 and 2 markers on linkage group 7. LD blocks are determined using haploview, and these 2 markers are in high linkage. These 2 markers are in high linkage and they are forming a linkage block. Here also we observe a very large block where the markers are highly linked, and a breeder can, if a breeder want to select some particular trait which is coming in this region, he can, instead of using all the markers, he can use only one marker from this region to select the trait of interest. In summary, we have used 1872 markers for 1872 marker data and heat and drought tolerance data for establishing marker trait associations. 3 subpopulations were determined using structure. We could find 583 significant marker trait associations. 100 seed weight was showing maximum number of marker trait associations, and these marker trait associations were explaining about 38.3% phenotypic variation, and the marker trait associations and the marker effects will be useful in selection of genetic material for genetic gains. I have already mentioned all the collaborators on this project, and I am also thankful to these people who are also helpful for me and all colleagues at Applied Genomics Lab. And finally, the most of this the project is funded by Generation Challenge Program and ICAR, and I am thankful to my supervisor, Dr. Varshni. Thank you.
Thank you very much, Mahendra. So a few things from me that we can open this discussion. You forgot probably to include Shesha Sai because for the carbon isotope discrimination data. This was from Shesha Sai. And of course, there may be several other questions. And so this is probably the preliminary analysis. It still needs to go to several rounds of the checking up, etc. because I could figure it out some errors kind of thing. But anyway, so we are now the floor is open for the discussions. And yeah, so who would like to start?
Just I was checking your grouping with perhaps dark markers where you find
You need to go there. Just
and white type going with three types.
Yes, sir.
Yes.
Yes.
That's based on dark marker.
Yes, sir.
Because our assumption is that in most cases, when we deal with the 46. We did find that, of course, there are two broad groups, basic personality and physical there. And wild type are slightly separate. Wild type, at all, they could go based on the origin and wild to cultivate. They should be basic type rather than perhaps the marker you have used are maybe partly responsible for that kind of distress. Evolutionary, we should not expect it to. We can discuss it later.
Yeah.
Just another thing is that this yield, maybe I heard it wrong. You said something that
Per kg per hectare.
so that's total yield. Average yield of India maybe 100 kilo, maybe less than that. Yeah. So this is the in relation to the experimental yield, isn't it? Yeah. We have to make clear. For experimental yield levels are 5 tons, it could be okay, but it will be experimental yield levels are only 1, and out of that, if a single marker is 762, I'll check my figure. 76 kilo I can think of, but certainly not 760.
So I think
Too big a figure.
no, I mean that was also a bit surprising to me, but in my opinion, but we need to check the data. So for instance, the data set which has been used for this analysis, in those data set, you may have the yield variation from something to something higher. So maybe you can have sometimes, I don't know, maybe this is just my assumption, 3 tons per hectare or 4 tons per hectare. So probably this particular allele is responsible to contribute to have the difference. So like for instance, in 2 different lines, you got 1.5 kg, another line you got, or in the other allele is associated with the line where you are having 2.2 kg, 2.2 tons, the difference is 768. I don't know. But if you can have.
No, because what happens if certain sequence data we have used? Was it consistent for all certain sequences? Yeah. Also, because if we take the average, can we really average out the data? Because some of these would need further analysis, whether we can really do the combined analysis.
So in few cases we had to throw some data set because of this reason that they were not comparable to those things. You want to say something, Abhishek?
If they were not, whether data was edited at this scale, because if data was not edited, then you would have this kind of discussion.
I think this is Mahendar, you want to add something? Otherwise, questions were asked to you. We couldn't speak.
Regarding this yield, we will relook again into that because I have checked the raw data also while I was talking to Ramu today morning. So he has also pointed out same thing. So I will once again check the data and so for this trait whether we should go for a combined analysis or not. So we can.
What is upset? You would feel very comfortable on that one, right? So yeah, so he says that this data set you can do the combined data set analysis, right?
Then I where I had a physical method going after 0.03 really does not.
Yes, that was the maximum positive contribution that we have observed.
What I mean is that biologically 0.03 seconds.
Yeah.
Yes. Really significant represented substantial increase into that.
Well, I don't think that there is an issue with that. But you want to say something first?
So all the markers that are associated with root length density, they were having some positive effect around only 0.03 centimeter per centimeter cube.
So there are two different kind of analysis when you do. So generally, majority of time, the plant breeding and genetics community are influenced by those linkage mapping based QPL analysis. So what happens in those cases? So if one parent and the other parent, they are generally the contrast, and then you have the mapping population. So then QPLs, what you identify, we always feel very happy when we say the marker is going to be 30%, 20 or like that. But we should remember that this variation is just the variation of the difference between two lines, and many times this is overestimate of the QPL effect, etc. But once you go in this kind of data set, when you are having a range of the germ plant with different kind of the genetic background, and in case if you are not finding something, this means that well, there is not. I mean, if you go for the cross from this one, the same marker can give you the 35% phenotypic variation. So these are, but then those association mapping analysis, there are two different components, and plant breeders and statistics they do better than me. So two things. One is the probability, the p-value. Yes, otherwise you can find the thousands, and then generally, even in the normal cases, even the QPL analysis, p-value 0.01 or 0.005 is very good. But here, you have filtered out. So whatever you have now, this is highly, highly significant. And the second is the contribution. That is these areas, new areas, so people have not done much work in this. People are learning now, for instance, the maize, and even this another question of the marker. So in chickpea, based on the ND stuff, we think that this is okay, but still I feel that we need to have large number of markers on the ND. Now the maize community, when they are doing this GWAS kind of analysis, they are better than maize because in maize you will ND decay very fast. So their assumption is that even 50,000 markers are not good enough. So they are going in the millions of those data points kind of thing. If you have that kind of data, because majority of the time we talk, we feel very happy when we say we have done association mapping. However, we should realize that with what kind of marker data set, how many markers, whether this was directly related to those LD. I don't know that, for example, this is my feeling, you need to check the SNP marker which you mentioned associated with the yield. This is not from the genome-wide skin markers, rather this is from probably the candidate gene based markers. Again, even those analysis were having two different kind of approaches. So you will, you see, if you are using a genome-wide marker, then you will have the possibility for 0.3%, 5%. You will have hardly any marker associated with more than 10%. When you go for the candidate gene, then you are having highly associated with the E7. So even so, you can generate data, etc. And then Mahendar needs to work very hard on this topic. So interpretation of this data is also very important. We need to see the or some people not. Earlier you were planning to give the seminar on some other topic. Then started to talk on this topic. But anyway, So yeah, so these are the issues. So in association mapping, so in base, and if you have that kind of marker density, then you will have markers associated with the 20% phenotypic variation. Going at each and every. Anyway, Ramu, you would like to answer the question, or you would like to ask the question?
I just asked about the question related to the gene-based markers.
So Ramu, before you ask, you also mentioned that in sorghum you did this kind of analysis.
Yeah.
How many marker data set you used?
I have used 265,000 markers to use it.
And then when you have these trait association, what was the maximum value of the phenotypic variation?
Variance, we have
This is maximum, but in general you will be having 5%, 10%.
10 is the
So hardly you will have 1 or 2, which is very on the top, and this is basically sitting in the gene, which is directly associated with the gene. So these kind of analysis can give you an idea, man, that try to put this thing in one picture, that you land in the genomic region, and then you start to ask, well, in this region, how many other genes I can target and sorry. Okay, Mahendar is answering or? Okay, sorry, yeah. But Ramu, you asked the question.
Yeah. You said we have used 113 gene-based SNPs, right? So these gene-based SNPs, you find one gene having associated with a trait. In addition, are there any traits are affected by these genes which you used? Yes. So if yes, then is there any markers associated from these 113 names of any of the traits?
So we have 10 gene-based markers. Out of these 10 gene-based markers, we had SNPs, 113 SNPs in these 10 gene-based markers. So only 2 genes, ACER and CAP2 promoter, were showing significant associations with different traits. So they are showing about with 10 different traits. I have not presented that here. So I can.
So there is a there are.
Yeah, I can talk to you with the help of that results.
What happens in many cases? So now when you are having those CAP2 kind of gene or kind of basically there is a transcription factor. So the variation does not mean that at one place this controls the expression of several genes. That's the reason that you will find such kind of gene or SNP associated many times with the different traits. Well, so any other question from some postdocs, students, or only you ask the question to them.
No, we consider some p-value. We take a threshold and then consider if the marker trait association is significant or not based on the p-value, not any phenotypic variation.
You had a question. Also, you are using all the markers for the cube. Or they need to study genetics as well. Yeah, so that's why I'm asking. So if I know, I can ask. Yeah.
So
first you understand the question, then answer.
What is the difference between the genomic selection and genome-wide association?
And you understand both of them?
I have little
Okay,
idea.
so whatever you have.
Maybe, sir, you can.
No, well, Dr. Patancheru already asked me not to speak. This is not my presentation.
So, in case of genomic selection, we have taken the historic data and we have developed a training population. Based on training population, we determine the genetic GBE values, the breeding values for a given trait, and then select lines based on that value in a population.
GBus. Most of the time when we use GBus and genomic selection, idea is to be different. Genomic selection, our idea is to identify
line based on the marker data may be significant, not breeding value of that line. But here in association mapping, we are more interested in identifying the markers which are significantly associated with the trait. So actually, they are two different things. But of course, as is mentioned, methodology is quite similar when you come to the more or less nowadays also converging. Like many of the increasing number of markers, people are using Bayesian markers for both of them.
I will ask here if some of you understand what is breeding value as such of an individual. Here the breeding value of line. Breeding value of an individual is that if you have 1,000 individuals in a population and you have one individual, you want to know what's the breeding value of this. So what you do is you have the population being in classical biological view and then see where this with all these 1,000 individuals and 5,000 the variations and then mean of the spread variations from the population mean will be the breeding value. The breeding value is always associated with the that sense it is. Here when we talk of breeding value, it is the breeding value of the value associated with the marker. And marker value, the value associated with marker.
Basically, you are saying that that would be the GBV, genomic estimated breeding value. When you calculate that breeding value based on the markers associated with that one, then GBVs, if you do based on the phenotypic data and do this additive kind of thing, then this is normal breeding, right?
So you need 5 comments.
Okay.
Breeding value is coming for particular
line.
In this case, marker.
No.
It is always for individual. Say breeding value is always for the individual in relation to population. How much it influence the change in the mean expression. Say if you say the breeding value, you say alpha 1 minus alpha 2. So alpha 1 is if you in the entire population, between a leading change with it, and then you get something or reverse alpha. The difference of these two is your breeding value.
This is hard for statistics, but as a layman language, that you have been given 100 lines. So which line breeder should use for making the cross? They can have, is that right? So then you can have the better genetic data. And then in breeding program, traditionally, as Dr. Baba was saying, that people were calculating those breeding values based on the phenotype. But now in the genomic selection, that in some of the lines, and that there are also caveat as well, what type of lines you are having the phenotypic data and you have done the marker profiling in such a way that you have covered those regions. So basically, you develop the models, and the models can be developed based on one population. And based on the markers which you identify, you develop the breeding value for the other population. Then you start to call it GEV, genomic estimated value. Then try to identify the line that well, and these breeding values there is that 0.020 like that up to 1.0, right? Maximum. Yeah. The same thing with GBV, so you are having 0.8, 0.7. Okay. Ramu, your question?
Just a comment. The Genome-Wide association and genomic selection both are integrated in a tool called GAPIT, Genome-Wide association and the prediction integration tool. This was recently published in Bioinformatics Journal, so very interesting.
And it's really downloaded.
Yeah, it's a very freely available from Cornell-based genetics site.
Okay, anything else? Tushar, you want to add something? Was comment? Okay, so there is no question, then probably we can close the seminar. And I would like to thank Mahendar once again, and thanks to all of you for coming to the seminar and having the very useful and fruitful discussion. So thanks once again for everyone.